Дякую, що прийшли на лекцію. У нас сьогодні рідкісна удача послухати представника американської школи алергології і імунології, професора Лоуренса ДюБаскі, який входить до десятки найкращих спеціалістів з астми і алергії в Америці, в США. Він великий друг України, він дійсно дуже багато робить для України, він дружить з конгресменами, які в тому числі ухвалюють рішення про надання допомоги Україні. Він залучений до цих процесів, він був в нашому університеті 17 років тому, але тоді не читав лекцію, але бачите, час спливає, все вертається на круга свої. І today, let me welcome Lawrence DuBuske. You see a lot of the... Лекція буде англійською, ви всі це пам'ятаєте, так? Я сподіваюся, I hope you will understand English perfectly. Anyway, we have the slides and you can read the information from the slides. Once again, I'm very glad to welcome today Clinical Professor of Medicine, George Washington University School of Medicine and Health Sciences from Washington, D.C., Faculty, Harvard Medical School, former member, Allergy Fellowship Training Program Director, Brightman and Women Hospitals. This is all previously held positions. Affiliate physician, George Washington University Hospital, Director of Immunology Research Institute of New England, which we are collaborating with all of these years. Founder of IRIN, past president and current historian of interasthma, distinguished fellow, past speaker of the House of Delegates, American College of Allergy, Asthma and Immunology fellow, American College of Physicians fellow, past chair of Practice Standards Council, American Academy of Allergy, Asthma, Immunology, past president, current treasurer of American Association of Certified Allergists, consulting physician, UMass Memorial Health Alliance, Leominster Clinton Hospital, Leominster, Massachusetts, USA, and this all is Lawrence DuBasky, professor. Please welcome him. And now, dear Lawrence, the floor is yours. Thank you very much for visiting us.
First off, I want to thank all of you for coming and agreeing to listen to me and hear what I have to say. I want to thank Vika for inviting me. This is my first time in Vinica. I first came here about 17 years ago and have been collaborating with this university through Vika for nearly 20 years. So a little bit of my story. Oh, this is a typical American thing, disclosure. You say, whose work are you presenting that may have pharmaceutical interest? And I am a consultant for a company called Allergy Therapeutics, and I'll be discussing some of our recent research that actually may eventually come to this university, we hope. First off, I begin with my parents. My mother's family immigrated from what is now Lviv Oblast. My mother's father came from Uknev, her mother came from Sanak, and the family immigrated around 1907 from what is now Ukraine. My father, Stanislav Shaborsky's father, was from Krakow, and again, my mother's parents were from Galicia region. Here's my grandmother, my mother's mother, Katarzyna Siganik-Szanovska, and she was from Sanak, again in Galicia. This is a photo from around 1905. Here's the wedding of my grandparents in New York City, Katarzyna Siganik and Yosef Shanovsky. Yosef's family was from Uknev, which is in Lviv Oblast. They didn't take long to have a big family. This is a photo from about 1920 where they already had 8 children, and my mother is sitting on her mother's lap in this photo. So this is my connection with Ukraine. It's a genetic connection. It's a family connection. My mother's first cousin, her mother and my grandfather were brother and sister. Stanislava Bunkowska Zemyevich. She was from Uknev in the Lviv region. She lived to 101 years old. And here she is with her son Jacek. They moved into Poland and are living in Dukla by the Ukrainian and Slovak border. Here's my cousin Jacek in Dukla in 2016. I first came to Ukraine in 1993. My initial relationship was with the Tuberculosis Institute in Kiev. And here I am in the front, look a little different with the brown hair, with Nikolai Kovalenko, who is a professor from that institute, and Alex Markov, who was my initial contact in Ukraine. Alex's brother, Dmitry Markov, was in the Kuchma administration and was ambassador of Ukraine to the Netherlands. I met George Dronik in the mid-1990s. Professor Dronik from Bogomolets founded the Ukrainian Allergy Society, UTAI, in August 5 of 1987, and I have had some association with this organization since it began. Over the years, my nonprofit foundation, Immunology Research Institute of New England, has supported a number of scientists from Ukraine. Here's Igor Kaidyshev and Volodymyr Biloglazov at the European Academy of Allergy and Clinical Immunology, 2011 in Istanbul. Here's Igor Kaidyshev, who's now president of UTAI. He's from Poltava, at the European Academy of Allergy in 2013 in Milan. Here's Professor Valentina Chopyak with Igor in Milan at that same meeting. And here's someone we know. Here is Vika Rodenkova with Igor Kaidyshev at the European Academy of Allergy, Milan 2013. Here she is once again. This is at the Immunology Research Institute of New England Symposium with Igor Kaidyshev and Leonid Titov, who is a professor from Belarus. Vienna 2016, Igor Kaidyshev and my close friend Yuri Basyuk. Yuri was a professor at Bogomolets and then later the Shupyuk Institute for Postgraduate Medicine in Kiev. He now lives in the United States and runs quality control for medical research at University Hospital in New Orleans. He's one of my closest friends. I've known Yuri since he was a medical student, like I think many of you are, in Crimea at the Crimean State Medical University. We met in 2000, and I had the honor of mentoring him, and he decided to go into this field of allergy immunology. Here is European Academy of Allergy Clinical Immunology 2022 in Prague. This is Professor Yeryomenko, Galina Yeryomenko, and Galina Koval from Kharkiv and Chernivtsi. I met with both of them over this past weekend as they came to visit with me here. I brought a contingent of 5 professors to the American Academy of Allergy, Asthma, and Immunology, including Professor Rodenkova. Here's Professor Galina Koval, Galina Yeryomenko, Valentina Chopyak, and finally Professor Lyshchuk-Yakimovich, Kristina Lyshchuk-Yakimovich, both from Lviv. And here we all are together in front of one of the posters of our collaborative work. I've been helping produce posters for my collaborative scientists in Ukraine for the last 20 years, and here we all are. Here I am with Professor Yeryomenko in front of her poster. And then we came to Washington, D.C. It was a stormy day. We drove in a snowstorm from Philadelphia to D.C. There were some questions if we'd ever make it. In fact, there were some questions if we should turn around and go back to Philadelphia, but we persisted. And here they all are in my office at George Washington University with a selfie taken here by Professor Kristina Lyshchuk-Yakimovich of the group, and you see Vika front and center. And here we are in the hotel room where I was in Philadelphia as they brought me a flag from Ukraine, which I now have displayed in my office. And finally, I took them to the White House, but I didn't expect that the White House would be in a cage. But our current administration decided to put a cage area around the White House so you can't get close to the White House now. You can only get about 100 yards from the White House, but they're all here a day it was snowing. Well, I'm on a faculty at George Washington University. I've been a professor of medicine since 2010, and I've gotten to know 5 congressmen. We had 1 Supreme Court justice as patients. And this is one of my good friends who has also been a patient, Congressman Brendan Boyle from Philadelphia. He's a Democrat and a strong supporter of Ukraine. And here he is in my office in Washington, D.C. I had Congressman Boyle on the phone with Professor Bisyuk, who was in Kiev at the time in the early days of the Russian invasion. And Professor Bisyuk talked with him and also with Congressman McCall, who I'll show you next, and I think helped cement their support for Ukraine. Here's Congressman Michael McCall, Republican from Texas. He led the initiative to get the $60 billion aid from the United States to Ukraine in 2024, April of 2024. Here he is in my office, and here he is giving an interview after the Ukraine aid bill passed in 2024. And I want to point out, I'm wearing today the same pin that he has on his lapel in his photo. I gave him the pin the day before the vote, as he and I were together the day before the successful vote for the support of Ukraine. So it's been a pleasure to be a part of Ukraine being recognized by Congress in the United States as worthy of support, and I hope that America will continue to support Ukraine, although we have some troubling times right now in our country, but like all things, this too will pass. Now my lecture. I've been involved with allergen immunotherapy for many years. I first got into this field in 1976. So you're looking at someone who's been involved in this field for 50 years. And after 50 years, I hope I know something, but it's taken a while. Allergen immunotherapy, allergy injections are the way to desensitize people who have inhalant allergies. But there are problems. Safety, adherence, efficacy, long-term benefit are all questions. And part of the answer to these questions may be the approach I'll be discussing today, which is to modify the allergen in some way to make it safer, decrease the length of treatment to a short course, and add an immune stimulator called an adjuvant in order to get a bigger impact. And one example is something I'll be discussing today called MPL. Allergoids are modified allergens. What you do is you polymerize the allergen using glutaraldehyde. This is a long-term solution for safety. The glutaraldehyde binds to epsilon amino acids in the protein, cross-links them, and forms a polymer. The initial work in the United States and the seminal work in terms of publications was done by Professor Roy Patterson at Northwestern University in Chicago in the 1970s with polymerized ragweed. Subsequently, a lot of work in this arena was done by Alexander Babakin from the Russian Federal Institute of Immunology in the 1990s. He passed away a few years ago, but he was a close friend and colleague, and he developed many polymerization techniques. The end result is we enhanced safety and gave less risk of anaphylaxis. So this is how you make an allergoid. You take an allergen like Vika works with pollen from trees or grass or ambrosia, which is ragweed. You extract it, you filter it, you get a natural allergen, and then you add glutaraldehyde to it to form a polymer called a modified allergen, an allergoid. The advantage of allergoids is you reduce reactivity to immunoglobulin E, shown on the left here, but maintain reactivity to immunoglobulin G of the allergoid, and you dramatically reduce allergen skin test reactivity. You preserve T-cell epitopes, but you lose immunoglobulin E binding epitopes that cause allergic reactions. And this is the man who was my mentor, Professor Roy Patterson. Again, I met him in 1976 at Northwestern University. He introduced me to allergy, and I just say to all of you who are students, mentorship is critical, and having someone who can guide you in your career, early in your career, is extraordinarily important, and he is why I'm here today. He published work in the 1970s and the early 1980s showing that when you polymerized ragweed, ambrosia, you could enhance the safety and allow for effective immunotherapy with just a few injections and with essentially no risk of anaphylaxis. This article was written together with the senior author, Albert Scheffer, who later was my mentor at Harvard University. In his study, it was 81 patients they looked at. They gave a large amount of polymerized extract over 15 weeks, and what they saw, which was critical, is that there was no anaphylaxis. Here you see the efficacy. The folks who got the polymerized extracts are in the middle curve. The top curve are people who did not, who got placebo. And you can see a reduction in ragweed-associated symptoms. And a critical thing was, again, no anaphylaxis. They then did the same thing with grass, and they did grass polymerization and published this article that showed with 13 injections, no systemic reactions occurred, and they gave a huge amount of allergen, 72,000 PNU, with no systemic reactions. This technique developed by Patterson was later taken up in Moscow by Alexander Babakin from the Federal Institute of Immunology. I know that talking anything about Russia today is probably not a good thing, but he was a very close friend, and once upon a time we could all be friends, and I hope one day that will happen again. He developed polymerized allergoids and monomeric allergoids in animal models, but showed that you needed to adjuvant them. You needed to add an immune stimulant to get an effective response that would tolerize. And he developed the concept of modified allergens with immunostimulation, and in fact he worked in my lab in Massachusetts periodically for about a 15-year period, bringing modified allergens from his institute to my laboratory, where we studied basophil histamine release using basophils from American allergic patients. So it was truly a joint collaboration. I'm next going to describe the product that I've been involved with more recently from the company Allergy Therapeutics, which uses two adjuvants and a modified allergen, microcrystalline tyrosine and MPL, a monophospholipid A. I'm also going to describe something that's absolutely cutting edge, which is a way of modifying the allergen by not polymerization, but attaching it to a virus-like particle so that the allergen can't cross-link immunoglobulin E, and again adding an adjuvant, in this case E. coli RNA and tetanus toxoid, to develop a strong Th1 immune response. And that is a product that I'm also involved with development of, which we hope will be the first food vaccine ever developed. And this could revolutionize treatment, and it's probably the most important thing I've ever been involved in after 50 years, finally something significant. This is monophospholipid A. It's an immune stimulant. It's currently owned by the pharmaceutical company GlaxoSmithKline. It's derived from Salmonella minnesota. It's a derivative of lipopolysaccharide or endotoxin, and it induces a strong Th1 immune response by interacting with toll-like receptor 4. Stephen Durham from the United Kingdom did some of the important work showing how this was working. This is a publication from 2006 showing that this adjuvant promotes a Th1 immune deviation. What he did was take mononuclear cells from 13 grass allergic subjects, cultured them with grass pollen, then looked at what happened when adding an extract without monophospholipid A of Timothy grass, and he looked at proliferative responses and cytokine production. And what you see is when you use the monophospholipid A, interferon gamma goes up, interleukin 5 goes down, and what you end up with is a massive effect on the cells that's based on interleukin 12 generation, which is a T helper 1 cytokine. If you take interleukin 12 out of the picture, you lose the benefit. So what you see on the left in the white is with interleukin 12 out of the picture versus in the picture on the right shown where you've neutralized the interleukin 12, where you don't have that benefit for gamma interferon, interleukin 5, and you lose the benefit of this adjuvant. So what the Durham showed is that this is an interleukin 12 dependent T helper 1 immune deviation induced by monophospholipid A. GSK has taken this to the marketplace by combining monophospholipid A in an adjuvant system with something called asapanin, which also drives Th1 immune responses. And there are now 2 commercially available products. One is a virus for respiratory syncytial virus. This vaccine against this virus has monophospholipid A plus saponin, and a vaccine against herpes zoster virus. Now I'll show you just briefly some of that data. It was shown that this MPL AS01 adjuvant vaccine gave a strong interleukin 12 response, similar to what I showed you with Durham. Interleukin 18 and interleukin 12 were both driving the immune response with stimulation of monocytes and induction of an inflammasome. And the inflammasome that you get at the site of the vaccination is part of why it works. There's a little pain. For a few days you have a sore arm, but you get a remarkably effective immune response. And here is shown the most recent data. This is from New England Journal of Medicine for respiratory syncytial virus, showing that there was an immune response that not only lasted 1 year, but 2 years. In studies that were done, they showed reduction in lower respiratory tract disease after immunization with this respiratory syncytial virus fusion protein MPL vaccine, and they showed the benefit was at least a year, and in fact they've now shown the benefit extends over 2 seasons, and they've actually now shown 3 seasons with just 1 immunization. So 1 immunization can protect against respiratory syncytial virus using this adjuvant vaccine for at least 3 years. And here's the study that looked at 1 year, then 2 years, then 3 years, and showed that there was protection over 2 seasons, and they now have extended it for 3 seasons. The herpes zoster virus immunization with the same immune adjuvant containing MPL has been shown to give protection for 12 years with just 2 immunizations. Here's some of the data showing that there's an antibody response and a cellular response, a T-cell response that lasts at least 3 years. And the long-term protection clinically goes for 10 years. This is data showing over 90% protection over most of that 10-year period. It declines to about 80% at the end of 10 years. And what's critical here is that you see an immune response in terms of antibody response and cellular response that will last 8 to 10 years minimally. So taking this monophospholipid A, it was decided to create an adjuvant system by combining it not with the saponin, but with something called microcrystalline tyrosine. And this was developed by Alan Wheeler on the top and Stefan Waronecki, who's a Polish scientist who immigrated to the United Kingdom, where he worked with Alan Wheeler. Ethan Allen Brown from Boston first described microcrystalline tyrosine. He was a physician from the UK. He was trained in London, immigrated to Boston, and practiced allergy in the 1950s and 1960s in Boston. When I first came to Boston in 1981, working at Harvard Medical School, I actually inherited some of his patients, so that's my connection to Ethan Allen Brown. He was one of the founders of the American College of Allergy, Asthma, and Immunology. But he published about microcrystalline tyrosine that it was a short-acting depot that could act as an immune adjuvant Unlike alum, it was fully metabolizable, and it only stayed in place for a few days and gave a very strong T helper 1 immune response. This is what microcrystalline tyrosine looks like. It's actually crystals of the amino acid tyrosine. And it can bind to the MPL and bind to your allogoid by essentially electrostatic forces that are beyond my capability of describing. They're called chi-psi electrostatic forces that binds the allergen to them. It's been shown in a model system using ovalbumin that tyrosine plus MPL gives a very big boost, shown in the highest bar, of allergen-specific immunoglobulin G, and shown here in green, IgG2A, which in an animal model is a Th1 response. Much bigger than you'd see with, for instance, alum and MPL. So I was fortunate to be named the lead investigator in 2007 of a study that was at the time the largest grass pollen allergy study ever done. We had 1,000 people in it. It was an ultra-short specific immunotherapy study using modified grass allergen combined with microcrystalline tyrosine and MPL. It was 4 pre-seasonal injections. The dose we gave was 5,100 SU, which was an in-house standard. And we did it pre-seasonally, 4 injections before the pollen season in 2008. And we showed a significant, highly significant reduction in combined symptom medication scores with the people who got the grass immunotherapy compared to placebo immunotherapy. And we showed a 24% benefit over placebo and a big rise in allergen-specific immunoglobulin G and no anaphylaxis. So we met everything we wanted to show. Tony Frew, who is past president of the European Academy of Allergy and an immunotherapy innovator, and I'd like to show him because Tony and I brought the first international allergy conference ever to Ukraine in 2000. Me with my foundation, Tony with the European Academy of Allergy. We came to Kiev for 3 days, then came to Crimea for 3 days, and that's where I met young professor now, Yuri Bashuk, who was a student at Crimean State Medical University. So it's very interesting how all of this sort of ties together. But Tony was a great friend who unfortunately tragically died a few years ago. Tony did a reanalysis of the data from our study and showed if you compared our results to the peak symptoms of placebo-treated subjects, you got an even bigger response, a 26% response, as opposed to the traditional way of looking at it, which was the peak pollen count, where we had a 24% response. So compared to placebo-treated subjects, at their worst, we were much better in terms of reducing symptoms. A few years ago, this man, Peter Jan de Kam, came to Allergy Therapeutics. He's a statistician. He had worked at Merck and Schering-Plough. And he really took this work to the next level. And I'll show you work that he did and his predecessor, Professor Tim Higginbotham, who was there for a few years running clinical trials before Peter Jan. What was found was we had the wrong dose to get maximum efficacy, but we had never dose ranged this product. So a dose ranging study was done. We found the optimal dose. We then took it into clinical trials, and ultimately, October of last year, it got German regulatory approval from something called the TAV process, which is German regulatory process. No other immunotherapy vaccine has succeeded getting this approval, so we stand alone. So the dose finding trial was actually developed by Professor Tim Higginbotham, and what we did is we looked at multiple doses. We began with our 5,100 dose, and we went up to a dose of over 30,000 units. We found around 27,000 S units. We got the maximum benefit on conjunctival challenge, where you put allergen in the eye of someone who got these injections. And we saw that we got maximum benefit in terms of allergen-specific immunoglobulin G, and we had safety. There were no serious adverse events. So we took that dose, a new dose, a higher dose, a dose that was more than 5 times greater than I had used in 2008, and put it into an exploratory field trial, and I'm honored to be an author on this that was published in a journal, Allergy. What was seen here was an interesting way of analyzing things. We had what we call the conventional posology, where we didn't have 4 injections, but 6 injections weekly, and something called an extended posology, where we had 3 weekly injections followed by 3 monthly injections. We compared, would it be better to spread the injections out or just give them on a weekly basis? And this was partly from the input of 2 men I'll show you later who are Swiss immunologists who suggested looking at normal immune response, spreading things out may be helpful. So we did it either weekly or weekly for 3 weeks and then monthly for 3 months. And we got the greatest benefit, which is the lowest line here in green, using the monthly for 3-month approach. And you see on the top this extended posology where every parameter, combined symptom medication scores, peak season score, every parameter achieved a P level of less than 0.05. So every parameter we successfully hit. Taking a look at this, the combined symptom medication score, much greater benefit using what we call the extended posology versus giving it weekly, and the benefit was huge, 39.5% benefit. So now we were close to 40% reduction of symptoms compared to placebo. We also showed the greatest improvement in allergen-specific IgG, greatest improvement in rhinitis quality of life, and we had very high adherence and no significant serious adverse events. Again, this study was critical in developing the ideal way of administering it, the extended posology, and the ideal way of assessing patients. We then did another study after this study, and it was called the Resonate study, where we used a very interesting study model. The idea is we may need 2 seasons to study this, 2 years of grass exposure. But if we did really, really well after the first year, we would break the data, and if we achieved a P less than 0.014, not 0.005, so a much more stringent P, we would be considered to have success and not have to go to do it a second year. And so this is what's called an adaptive design study, a very unique design developed by Peter Jan de Kam, again, a statistician. So here's the study, our 6 injections, 3 weekly, then 3 monthly. These are the people who were in it. They were highly sensitized to grass. Their allergen-specific Ig was very high, 28 kilunits. And we showed excellent results in terms of reduction in symptoms and a combined symptom medication scores, greater than 90% adherence to the regimen. And these are the results: combined symptom medication scores, daily symptom scores, daily medication scores, all significantly reduced with P's less than 0.05. And quality of life improved significantly, and immunoglobulin G going up 6-fold, and that's consistent with efficacy. We had Professor Mohammed Shamji, who's incoming president of the European Academy of Allergy and Clinical Immunology, working with us as a consultant doing biomarker work. And what he has shown is that successful allergen desensitization is associated with a big rise in immunoglobulin A1 and immunoglobulin A2 towards the allergen, and a reduction in facilitated antigen binding of B cells to the allergen. And we saw all of these things consistent with successful immunotherapy in the Resonate study. We also had high compliance, greater than 90%. Keep in mind, conventional allergy shots that require 3 years of treatment only achieve about 15% adherence. So only 15% of people have success. Here we get 90% of the people adhering. And then we analyzed the 2 studies, the G306, G309 together, and in a meta-analysis we showed we were comparable to the results that you get with grass sublingual immunotherapy, which is FDA approved and European regulatory approved, with about a 20% improvement. And now we're in a long-term pediatric study where we're going to see if 3 years of treatment will give at least 2 years of benefit after stopping treatment. So this is where we are today in a long-term initiative. And as I mentioned, we just got German regulatory approval for this product October of last year. So very proud that this is the first immunotherapy product to meet this very rigorous German regulatory approval. So safety, adherence, efficacy, and we hope long-term efficacy will ultimately be shown. We know, again, MPL vaccines can give 10 to 12 years of benefit. We think one 6-shot course may give 10 to 12 years of benefit. Now I'm going to conclude by talking briefly about peanut allergy. This, unlike grass allergy, is a worldwide threat to life and health. 2% of the Western population is peanut affected. Every year people die from anaphylaxis, not only from peanuts, but from tree nuts, from egg, from wheat, from milk, from shellfish. So this is a big issue. And anaphylaxis is the symptom, and the symptom can be fatal. The management traditionally is just avoid the food, but sometimes that's just not possible. So you have avoidance. There's now proposed peanut powder that you'd have to swallow every day. Omalizumab, which is a monoclonal anti-immunoglobulin E antibody, has been shown to be successful, but you got to take it the rest of your life, and it costs almost $100,000 a year the rest of your life. Not terribly practical. So novel treatment would be to get an immune modulator, pharmacy-grade product, and get a short course, once again, with an immune adjuvant to allow people to be desensitized to food allergen. And these two men developed it, Thomas Kundig and Martin Bachmann, both from Switzerland. And they're the inventors of the virus-like particle, a nanoparticle, that we're using in this product. This is what it looks like. It's a virus-like particle derived from cucumber mosaic virus, and on the surface, we attach a recombinant allergen, and we space it such that it can't cross-link immunoglobulin E molecules on mast cells or basophils, and it can't induce anaphylaxis. It's ingenious. And also on the surface, we put tetanus toxoid, which acts as an immune adjuvant, an immune stimulator. So we have tetanus toxoid, we have the allergen, which is recombinant on the surface, and inside we have encapsulated prokaryotic RNA that gives a big TLR7, TLR8 immune response, leading to non-Th2 immune responses, whereas the tetanus toxoid gives a very strong memory T cell response that again is non-Th2, and together these two adjuvants should enhance efficacy, whereas the virus-like particle and the way the recombinant allergen is on it gives you greater safety. So we've taken this into clinical development. We're being helped by Imperial College in the United Kingdom and University of Bern. Again, the company Allergy Therapeutics. Johns Hopkins University is also involved. We've shown in animal models an effect with intravenous challenge of animals who were treated with this vaccine, a big reduction in anaphylaxis and a reduction in skin test responsiveness. Immunization against the single allergen protects against systemic challenge with whole peanuts. So we showed this recombinant peanut allergen would prevent systemic reactions to whole peanut, again, using animal models. So you go from the animals to the humans. We're now in a process of doing a study on adolescents, adults, children will also be studied. And what has been shown using basophil activation testing, dendritic cell assays, B regulatory cell assays, T cell assays, is that there has been what we would consider a protective response induced by this unique vaccine. And this is what we've seen. There's a big boost in innate response with dendritic cell 1 type genes, which are innate immune response genes. a big improvement in terms of IL-10, which are immunoregulatory B cells, and a big increase in interferon gamma and a reduction in interleukin-4 from T cells, consistent with an immune regulation. So we see reduced capacity to activate basophils, pro-tolerogenic dendritic cells, immunoregulatory cells induced, and reduced Th2 responses. So right now the PROTECT study is underway, where we're using healthy subjects first to assess tolerance, and then allergic subjects to assess both tolerance and efficacy. And the model system that we are working on has only had a few subjects, but we've shown safety on skin prick testing in peanut allergic subjects. 6 subjects completed with well-tolerated and skin text reactivity seems to go down with exposure, and that's shown here, with no systemic reactions. So far, so good looking at skin testing of allergic subjects. We've then done some analysis of allergen-specific IgG, and it goes up dramatically with this vaccine, and that should be consistent with a good immune response. 42% increase in allergen-specific IgG versus placebo. And what we see is an impact that we've had in terms of skin test responses, relative allergen IgE, B-cell responses, and we've seen safety with no anaphylaxis thus far. So the preclinical work seems to indicate efficacy. We believe we're going to have an effect with just 3 injections. Wayne Schreffler from Harvard Medical School just presented this in Philadelphia at the American Academy of Allergy, showing both an effect on skin test and IgG to allergen, and all the things that we'd expect with successful immunotherapy in terms of induction of a tolerogenic response. So in summary, this VLP peanut, at least conceptually, should work. It seems to be safe, and it could be a platform for any food, not just peanut. And we think that the strong immune response we're getting should allow us to tolerize people to peanut. And this work is ongoing. And we want to acknowledge all the people, Imperial College and Allergy Therapeutics and Harvard Medical School, Johns Hopkins University, all been involved. So here's the team involved. I'm a proud member of this. Matthias Kramer is on the left from Benkard, Pirion Dekam in the center with me, and Professor Moshamji from Imperial College. And on the right, you see Janice Lahady, who's also from Imperial College. And these are the people who are involved. And in conclusion, modified allergen immunotherapy, I think, is going to be the wave of the future. 6 injections, desensitizing people to grass. And I gave Vika encouragement here. Ragweed could be next. And we hope, as the unfortunate situation Ukraine is in now, hopefully ends soon, that we will be in Ukraine. I was in Trinity yesterday. Here today, Vinica. Ragweed is a big allergen here. We hope in the future to be able to bring modified ragweed clinical trials to this university. The virus-like particle is first in human vaccine for food allergies. Again, I think the most exciting thing I've ever been involved with. I want to thank you for coming here today to listen to me, and Slava Ukraini.
Thank you very much, dear Lawrence. Дуже дякую вам, як слухачам. Я не знаю, правда, чи багато ви зрозуміли, тому що Лоуренс говорить швидко, і чи всі студенти тут знають імунологію, і викладачі, сподіваюсь, так. Суть лекції, те, що сьогодні ви прослухали, це фактично те, що каже Лоуренс, cutting edge, тобто найпрогресивніші дослідження у сфері лікування алергічних захворювань. Це алергенна імунотерапія, коли алерген природній модифікується спеціальними ад'ювантами і додається інші речовини, які можуть пригнічувати імунну систему. І це те, що зараз є в імунології, алергології якби найбільшим проривом і те, над чим працює світові такі наукові уми. І ви сьогодні, я розумію, можливо, хтось не доріс ще до цього, але ви, сподіваюсь, будете згадувати цю лекцію як одну з лекцій дійсно провідних професорів, дійсно провідного рівня, провідних університетів світу, тому що Лоуренс багато років працював в Гарварді, тепер він працює в університеті в Вашингтоні. І ми, звичайно, дякуємо йому за цю лекцію. І найголовніше, ми дякуємо за як підтримку України загалом, так і нашого університету, тому що наприкінці лекції Лоуренс пообіцяв співпрацювати з нами і надалі, і в тому числі, бачите, і незважаючи на таку ситуацію, яку ми маємо в Україні, якраз з питань розробки наступних ліків проти алергії на амброзію. Якщо ви знаєте слово амброзія, знаєте алергія до амброзії, то якраз над цим, я сподіваюся, наш університет разом з професором ДюБаскі буде працювати в наступні роки. І це також та частина, яка єднає нас зі світовою наукою і зі світовою клінічною практикою. That's so, Lawrence, thank you once again, and thank you for coming today, and we all appreciate your coming too, the coming of our audience and attention. Дякую вам щиро. Дякую.
Thank you so much. It's an honor to have been here. I want to salute all of you who are here. I can tell you, as an American, I was very proud when we gave the $60 billion aid to Ukraine in 2024. I remain hopeful that the politics of the United States will be changing soon and go back to supporting Ukraine. But I urge all of you to continue to fight, continue to defy, and continue to be resolute in defense of your country. This country, when it finally got its freedom after the collapse of the Soviet Union, allowed for the first time for the Ukrainian people to have their own nation on a long-term basis. I know after the first war, after the Russian Revolution, there was a few years when Ukraine surfaced as a country, but now, over the past 35 years or so that I've been coming to Ukraine, I'm amazed at the science here. Today, Vika and I went to the center of your city where there was a drone school, which I thought was absolutely amazing. And I actually took a video of it, and I'm going to be sending it to my friends in Congress. I think everyone in Congress in the U.S. should come to Ukraine to see the strength and the power of the Ukrainian people. I'm just amazed. I've gone from village to town to see all the memorials that are present. Every person who has died in this war is being remembered. It's stirring. And I want to salute all of you for your strength, your resilience, and the fact that it's due to all of you that we have a Ukraine today, and how Ukraine will survive is based on the work of all of you. As one of my friends said many years ago, Ukraine will live forever, and it will live in all of you. So thank you.
Я сподіваюся, ви зрозуміли. Тут Лоуренс дуже багато розповідав про те, як він захоплюється Україною і просив нас стояти далі. І сказав, що і він дуже захоплювався нашим містом, побачив сьогодні школу дронів і каже, що він теж дуже здивований тим, що всіх людей, які загинули, ми пам'ятаємо, і це все він розповідає в Америці і цю інформацію надає на самих вищих рівнях. Ну, давайте вже так, дійсно подякуємо Лоуренсу і дякуємо, до побачення. Якщо, може, хто хоче фото, думаю, можна зробити декілька фото.